All right, we're back again after a very short break. Today's speaker is Professor Leelavati Narlikar at IISER Pune. She's coming from the Department of Data Science. I think it's a brand new department, so I hope the students will bug you about that as well. So over to you.
Thanks, Sutika. Thank you very much for the invitation. Isn't it on? Yes. It is on. Okay, great. I was here last time, but I was online, so this is a much nicer experience. So thanks for inviting me again. I work at the Department of Data Science, and those words are not mentioned in the title. Okay, so the title you might think is a little weird because it's got computer science and biology. Usually these two are not taught together, at least. Can I just get a show of hands? How many of you had any course related to biology in the last year? Last year, first year of your, nobody, right? Oh, there is one, great. Okay, one or two. Well, I should add that I also had no biology course in my undergraduate program. So how about 11th and 12th? That's a bigger number. Okay, great. I didn't have it in 11th or 12th. So I picked something else. I forget what it was. So anyway, so what I'm hoping to convince you in the next 1 hour is that biology is very interesting, and many problems in biology can be solved using computer science and mathematics. Okay, and at any point in time, if you think during the talk something is not clear, you can ask, right? But of course, I don't know if I'll have the answer because my biology is also kacha. Okay, whatever I've learned is in the recent years. All right. So when you think of computer science, what do you think? What do you think of computer science when you hear the words? Code. Great. And biology also has this big code. Okay? It's called the DNA. Okay? It's often called, okay, let's see what's going on. It's hung. The pointer works, but the slide is not changing. Nope. There, now it changed. Okay. Yeah. All right. There. So DNA is often called code for life. And I think you'll notice a few similarities between the computer code and this code. Okay. So it turns out that cells store a lot of information, especially hereditary information, on DNA molecules. Okay? And the complete DNA content is called the genome. Okay? So you may or may not have heard this term. All it means is that it's DNA. Okay? And for people like me in the audience who didn't know starting off what DNA was, it's essentially a polymer. Okay? It's basically a repeating repeating set of units, which come in 4 different flavors: A, C, G, and T. Okay, you don't need to know the full form, just try to remember that there are 4 of them, and there's A, C, G, and T. And here's a single strand of DNA, okay, which, like I said, is a polymer. And in our cells, usually DNA does not occur as a single strand. Okay, it occurs as a double strand. Okay, and there is a pairing between what we call these units nucleotides in a very specific manner, right? So A always pairs with a T and a G pairs with a C. You know all this, right? Hopefully. Great. And you must have seen this structure, right? It's like a double helix. Okay. All right. So this is basic primer on DNA. Okay? And so in our body, okay, humans have 23 pairs of chromosomes. This is also something that I'm sure you know from school. Right? And if you just look at all the DNA in there, okay, what is a chromosome? chromosome is a portion of the DNA, genome, right? So if the whole genome is all the DNA in these chromosomes, right? And if you just look at all the DNA, the nucleotides, we have more than 3 billion letters, okay, 3 billion ACGTs. So that's a lot of code, okay? It's a lot of data or information. Okay? Is that clear? Okay, great. All right, so one of the big efforts in the last century was the Human Genome Project. The goal was to sort of sequence or find the sequence of this DNA, okay? So all the 23 pairs of 23 chromosomes, okay? And this was a big effort, and lots of different institutes, countries contributed to it. A lot of money went into this, okay? Many years. And finally, we had one version of it in 2003, okay? Probably about the time when you were born. Yeah, okay, all right. That sounds so old, okay. All right, so how did this happen? Okay, so if you think about it, this is the picture that you see under a microscope under very specific conditions of the cell, right? If you do something to the cell and you look at it, you might be able to see this, the structure of 23 chromosomes. But if you look at it in terms of, in a lab, it looks like this in a test tube. Okay, this is isolated DNA. So from this picture, right, or this test tube, how did these people come up with the sequence of the 3 billion letters? Does anybody know? No, because you don't have biology, right? Okay, great. Okay, so you'll learn something at least from this talk. All right, so it turns out that, let's go worse than biology. How about chemistry? No, okay. All right, so this is really very clever use of chemistry to find out the sequence of DNA. Okay, I have a picture on it, but in the interest of time, I'm not going to talk about it. Okay, but if you want, we can discuss about how you can sequence an unknown DNA. Okay, but basically it uses cleverly made molecules to sort of decipher what your input DNA sequence is. Okay, so have you heard of the polymerase chain reaction, PCR? Great. So it comes from there. Okay, there's a lot of science that went on after that, a lot of discoveries, after which we are at this stage where you can quickly sequence little pieces of DNA. Now, the key here is the little pieces of DNA. Okay, so this technology cannot sequence for you the complete chromosome. Okay, so the biggest chromosome, I think, is about 200 million in our cells. The total size is 3 billion, remember? And so what this does is it can give you a good accurate sequence for up to about maybe 100 or 300 bases, not beyond that. So how then do you get from this 300 or sometimes even lower, but accurate, fairly accurate sequencing technology, how do you get from there to this big DNA? Right? The question is clear, right? So you're starting off, imagine that you have this massive long DNA that you want to sequence. Okay? So when I say sequence DNA, I mean you want to find the sequence. Okay, I'm going to use this a lot, so just bear with me. That's the terminology, sequencing. Okay, so what you want to know is this, okay, from that test tube. What you can do are pieces of the DNA. So what you do then is you take many copies of your DNA, you either amplify in some way, or you just have multiple cells, so you get more copies. So you're not stuck with just one DNA molecule, you have multiple identical DNA molecules. Okay, and then what you do is you cut them up. Okay, because you can't do the whole thing. So if you start off sequencing, you'll just get till here, and you'll be stuck. Or beyond that, you'll get really bad quality reads, sequencing. Right? Question? Why does that happen? Why does it happen? Because this, so if I'll show you the picture later again. So it involves attaching different molecules and taking pictures okay, of what has been attached. Okay, so then this, as error creeps in, the error kind of multiplies over time. That's why it's inaccurate. But the imaging technology can tell you that, okay, beyond this, I'm not really sure what's going on. Okay, so it can tell you that because you have 4 different colors. And if you get some mixed color, then they know that there's something weird going on. Right? So you get a sense of, from here, your quality is going bad. Okay? So after you copy this, you have multiple copies, you cut it up. Okay? And now you can find the sequence or sequence these fragments. Okay? Everybody with me so far? Any question? Good. Right? So what you're going to get at the end of this massive sequencing experiment is just a bag of reads, a bag of little sequences. So this is also going to happen a lot. Okay, I'm going to call little sequences reads. Okay, this is also unfortunately something that comes from working with biology for a while, but just bear with me. These are interchangeable words. Okay, and I might call them reads. All right. So what you get is this bag, and what you want to get, yes.
What's the word were you using?
Read.
Read.
Yes. Yes.
So in the previous slide,
Mm-hmm.
when we cut the sequence,
Yep.
so we use the same sort of enzyme to cut all of it.
It would be random cutting. So it's just going to be sonication. You know what an enzyme is. It's cool. Okay. Yeah, no, you're usually they'll just cut it up randomly.
So when you're like sequencing it, won't you like sort of have a problem with
Those are also words you know. No, yes. So there are, so the chopping is random, and you can attach things to DNA, which might be separate, which you then use to sort of sequence the DNA. But it doesn't matter where the cut happened. Okay, unless you get really tiny cuts or really long ones, then you're in trouble because again, you're going to have these reads that are not very accurate. Okay. So what you get are these little sequences, and what you want to do at the end is to somehow arrange them, right? So that you're able to read out the real sequence, right? This is what you want to do really, because this is how what you had multiple copies and you cut them up, and you want to essentially make use of the fact that you're going to have overlaps. Right? The cutting is random. If the cutting happens at the same position each time, then it's useless because you're going to get the same reads. Yes. Because if you get just one, if you just have one copy and you cut it up, you don't know how they should be put back together. Yes. So this is like a jigsaw puzzle, and these overlaps are trying to tell you, in some sense, how they fit in. Right? So that fitting in information is lost if you just have one copy, because they can just attach anywhere. Does that make sense? That's the reason for doing the copying. Thank you for that question. I should have mentioned that. Okay, so you want to get back here, right? So this is the problem that you have. Pictorially, visually, you know what you want to do, but the first step is to now formulate it mathematically so that you can at least go ahead and get the computer to solve it for you, because this region, this DNA piece, is massive. It's not so tiny that I've shown in the picture. Right? So how do we think of formulating this? So what is the data that we have? The data we have is just this part, right? All these guys, not in this order. We don't know the order. Right? So let's say call them strings. Okay, little strings, little subsequences or sequences. So you want to find a string, G, that G stands for genome in this sense, in this case, so that every little string is part of that genome, right? That's essentially what you need to do. Does that make sense? Okay. So so this is one kind of formulation. So basically these are your S1 through Sn reads, the green part, and you want to get back your G, which is the red. Let's take a small example. Okay, suppose these are your reads. Okay, if you just do it naively and not look at overlaps, you can just, put them one after the other. This is one way of doing it. It still satisfies the graph problem. I mean, okay, forget about the title. The title will be explained in a bit. Okay, so this will still satisfy the question that we mentioned here, right? So what is missing? What do we need to put in there? Any idea? So do you realize that this is not what we want? Yeah. How many ways can you put the genome together like this if you had n strings? N factorial, great, right? There's so many different ways. There's no unique answer, first of all. But secondly, it's also not biology. No, it's just we did all these copying for a reason. It's not so that you can just add them one after the other. So what's missing? Can we change our formula? Can we add anything to the model form? Sorry? Pairs. No. So this is the nucleotides. No, there is no G in this. Is that what you're pointing out? Yeah, this is just an example sequence. Don't worry about that. Because if there were too many, then I can't work with it. So first I had only 2 A and T, then I added a C to make it look like a DNA. But that's a good observation. There is no G, but imagine there is one. Okay. So how do you constrain? What is it that you really want? No, there is no reference. My God, she knows all these words. Oh, that's good. No, we don't have a reference. So we're doing a de novo assembly where you don't know anything. Yeah. What's missing here? Do you think our DNA is going to be so long? No, right? So what kind of constraint can we add? How would you put that length? Imagine that the biologist doesn't tell you anything. Just tells you that I've cut it up, I've made so many copies, cut it up, and I'm giving you these sequences. That is true, but that you have been given. That you have. That's your data. You can even, that's in there. Okay, so I said that if you just make the problem statement like this, given these strings, you need to find a G that is a super string, right? And I just arranged those little pieces, 5 pieces in that manner, which you all agreed was not the correct way to do, right? Do you see that it's not the correct way to do? Because we are not using the fact that they're overlapping, right? And the fact that it was copied multiple times, right? So what is missing here? What do we really want to do? Is the answer going to be so long? We do, yes, but we don't know them. So how do you put it? No, you have, this is the problem. All you have are the sequences. So the barcode, you don't know whether you're putting it at the start. You chop it up and then you're tagging. Yeah. Yeah. Any idea what should you be doing? There's just one word you should be adding. It shouldn't be any string, no? It should be the shortest string that can do this. No, you don't agree? No? And that should take care of the overlapping part, that you will be forced to make overlaps because you want a short string, right? You could even add random DNA to this G. You could add a G and it would still satisfy this problem, right? The guanine, I mean, not, yeah. So in this case, the shortest string happens to be this one, I think. Yeah, okay. It's shorter. Yeah. So this is what you want to get to. And adding the word shortest helps you get there. Whether it's biologically correct is a different issue. Okay. Yes.
When we're chopping them up, it's like all different sizes. What if one sequence is like something that is repeating in the original DNA sample
yes.
you have? Then you sort of neglect that.
Yes, that's a good point. So if you have a repeating element, right, in reality, in your genome, she's saying you have something that repeats like maybe like you have a 10 bases, specific 10 bases that is coming on 3 times. right? If you use the overlapping business, you're going to just cut those out. Yeah. So there are other ways to deal with this problem. One thing is now the biologist will tell you approximate size. You can use that information, but this is, like I said, this is the first solution that we're coming up with. Okay, there are more advanced ways of dealing with this. Okay, so if you wanted to solve this problem, one way that you can do it is using graphs. Do you guys know what a graph is? You have vertices and edges, not the graph paper wala graph. Okay, it's a more abstract sense of a graph. Doesn't matter if you don't know. So we have nodes and we have connections. Okay, a graph is something you've seen, or a network, maybe you've heard this word, right? Okay, so for example, we use these all the time when Google is telling you the shortest path, right, from A to B. It's looking at regions that you have to go through, right? So this is essentially our nodes, okay, which are our strings. And you have an edge from a string S1 to S2 only if you have an overlap between the strings. So the suffix and the prefix, you know what suffix and prefix means? End and the beginning, match. Yeah? And they are weighted. So you have a weight associated with it as to how much is the overlap. If you have an overlap of a big number, then that has a bigger weight. Okay? Or if you have just one, then you have a smaller weight. So if you have an overlap of just one nucleotide, maybe that's not the best way of sort of connecting the two strings. Okay? So there are different graph algorithms that people apply to solve this problem. Okay, and I'm going to leave it here. This is just to give you a flavor of things you can do using maths and computer science. Okay, so let's move on. Any questions so far? Good. Okay. All right, so that was then, right? These overlap graphs and everything came long ago. And since then, both the technology, which has advanced because of advances in biology and chemistry, very important fields of sciences. Because of them, the cost of sequencing has really gone down. Do you know what Moore's law is? Never mind. Okay, so just showing you that it is the cost is decreasing really fast. You can see the scale. Scale is not linear; it's log scale, and it's even super log. I mean, super exponentially decaying. That's what it means. Okay, so the cost now has really come down because of these technologies. And what has happened as a result is that you can just sequence your genome with sometimes using what is called a reference, but even de novo. You can do all these things rather cheaply. Okay. So what you have because of this effort is, oops. All right. It's something called a genome, which is sort of represented in a genome browser. Look up the website if you're interested. So it has all the chromosomes and the nucleotides that we know that belong to each location. Okay, so you can go and feed any location on any chromosome that you want. Check it out. You'll see what DNA sits there. Okay, it's that good. And you can zoom out, right? You can see more nucleotides. You have the whole thing in there. Okay? And over time, with these advances in technologies and algorithms, both, they've managed to get better and better iterations. They're able to fix all these repeats that she's talking about. Exactly. Earlier, there were just these blanks where we don't know what is going on. But now we know a lot more about what's going on in the genome. Okay. All right, and this was a huge effort, and because the technology is here, you're not going to stop at the human. You're going to do lots of genomes, right? So just to give you a sense, human genome is this big, right? Giga comes from 9. Yeah. This here, do you know what organism this is? It's bacteria. Good, yeah. It's E. coli. It's a bacteria, which is smaller. Okay, as you might expect, right? The bacteria is really tiny and is a single cell, right? We have so many cells, maybe clearly more complex than a bacteria. You know what this is? Yeah. Okay. This one is bigger than us. Okay. So the question is, does this mean that an onion is more complex than us? Layers. Yeah. I didn't think of that. Sorry? I know. The onion is more, but does it mean that the onion is more complex? We know that it's not, right? Okay. So just going by size doesn't mean anything. We need to know what's written in the code. If you have a huge code, it can be very, inefficient, but you have a small, succinct code might be good. So just looking at the size doesn't always tell you much about the organism. Okay. So what then should we be doing? We should be trying to see what is really encoded in the sequence. What is the software really doing? Okay, this is what it looks like. So it's not very easy for the human eye to make sense of. So what are the parts that are important? Right? Why is this really considered code for life? Anybody has any idea what the DNA has? What kind of information the DNA has? Proteins, right? Information about proteins. Okay, so it has genes. Okay, so what happens is that the whole point of DNA, one of the points of DNA, so you should never say, in biology that this is what happens, because there are always so many different exceptions that keep popping out. So one of the things that the DNA does is has information about making proteins. Okay, have you seen a picture like this? Yeah. Okay, you have the nucleus, and then you have the outside cell. Right. So this is all the cytoplasm. You have the DNA that is sitting there. Turns out that the information on the DNA is transcribed and then translated to make a protein. Okay, I'll tell you what that means in just a second. I have another picture with some animations, so bear with my animations and praise them. Okay, all right. So here's the gene, okay, which lies on the DNA. When I say it lies on the DNA, it's just a section of the DNA. Imagine your DNA is a big book, and maybe your gene, this red gene, whichever one I have pointed out, starts from the 4th word in the 5th paragraph of the 18th page, and it ends somewhere else, right, later on. That's all it means, okay? It's all ACGT after all. The characters are same. This is the alphabet of our DNA, right? It's made of ACGT. And part of it is just coding for a gene. So what does coding mean? If it is going to be used, it needs to be first transcribed. Okay, it's transcribed into another molecule. DNA is a molecule, RNA is another molecule, which is kind of like DNA, but it has only one strand. Okay? And it is a copy. The only difference is that instead of a T, it uses a U. Okay, but I mean, you can just say that's a T. Okay? And the way this happens is a process called transcription, okay, which makes use of the really cool thing which we didn't talk about, was that our DNA comes in 2 strands, right? And it always comes in 2 strands. And if you think about it, that's double the information, the same information that you're storing, right? Because the information, once you know for one strand, you automatically know the other strand information. You know the pairing, right? So why are we storing so much duplicate information? The reason is for this, is for the chemistry to work out. So when you have the DNA, you want to make a copy, okay, of the DNA, and you make use of the base pairing. U means the cell, the polymerase makes use of this pairing to create a copy, right? So only when there is an A will it put a T there, and so on, right? So it will make a copy of this strand. Does that make sense? By reading this one. Okay, because the RNA is very much like a DNA with a few missing things. Yeah. Right. So right. So that's the use of that copying and the double information that we have in our body. Okay. So now this is a picture from this is a very nice book. If you have access to it, just go read it if you're interested in biology. So here's the DNA. Okay, that's the main DNA in your nucleus. And what you see here, do you see these strands coming out? Those are all the transcripts, okay, made by the RNA polymerase, and there are multiple of them. It's a gene that really is required. Many transcripts are being made, copies are being made again, sort of very quickly. So the first guy has already reached there and made the almost full copy. The second guy is here, third guy is, and then the last guy has just started the polymerase. It's a nice picture that shows this just popping out. So we are making all these copies of the gene. Okay. The next step now is the translation step. Okay, you remember transcription happened in your nucleus. Now translation is coming out after coming out. And this is where the protein is being made. Okay. So I like to think of this as a meal that you're making. Okay, so suppose your goal is to make this protein, which is, let's say, chapatis, something, or parathas. Okay, and you don't know how to make them. Right? So the recipe for that special paratha sits in some library. Okay, it sits in a nucleus. And you can't bring the book out. They're very fussy about that. All you can do is make a photocopy. So what you do is you go to the library, you look at the recipe that you want, you make that copy, photocopy, bring it out to your kitchen, okay? And then now you have to read this copy and make your protein. Okay, so these characters are the alphabet of our code. Remember, we're trying to connect this to computer science, right? So our DNA is ACGT, your RNA has this alphabet, and now protein suddenly has a lot of 20, okay, characters in there, in the protein alphabet. So information over here was easy, 1 to 1. Information here is trickier because you're going from 4 to 20. So you need more than just one nucleotide to give you information about which alphabet comes here. These are all amino acids, okay? But a different alphabet is good enough for us. And the way this happens is also called a code. This is called a genetic code. Okay? All right. So the way this happens is this is your RNA, right? That has come out and waiting in the cytoplasm to be read for the paratha to be made. And you have the ribosome who's hanging around there, starts scanning until it finds an AUG. Okay, that wakes up the ribosome and says, oh, this is the start of my protein. This is where now I start making the protein. And it starts substituting for every triplet one amino acid. Okay, so it's going to scan. Let's see. Oh, it finds the AUG, right? It's going to attach an amino acid that codes for AUG. Okay. Then it moves over to the next triplet, sees that it's a CUU, looks up and puts a leucine, and it goes on. Okay. And it goes on not till the end of the transcript, but until it finds one of the stop codons, which are these, which just tell it to stop. Okay, so it's just very, this is not the sort of ad hoc, right? This is what is happening. It is reading, right? Sometimes we think biology is just all ad hoc and, you know, we don't know what's going on. But this is a very specific code that the ribosome is executing with the help of many other molecules and making your protein that corresponds to the gene that was encoded there. Okay? Make sense? We're going to come back to this. The reason why I'm telling you this is we have to make sense of the sequence that we have assembled. Okay? So you stop there. Right, so this is our protein. And the proteins are, you know, get a lot of glory because they do a lot of things for us. Right? I don't have to justify why we are interested in proteins, right? Protein is a good thing to have. Okay. The many different types of proteins do many things for us. Insulin, for example, is one of them, right? You know, these are all critical proteins. They do a lot of stuff for us. Okay. So now let's go back to this. Now, can we predict the genes? Now that we know that DNA encodes genes, can we predict where the genes are? Okay, so what information do we have? We know this triplet information, which, by the way, is conserved all the way from bacteria, okay, to us, the same 20 amino acids. Do you see why we need 3 nucleotides? Would 2 have been enough? With 2, how many amino acids could you code? Only 16, because you have only 4 choices for the first one into 4. There would have been only 16 different amino acids, and if you had a stop codon, you know, you'd be stuck. 2 are not enough. 3, how many do you have? How many codons do you, how many triplets do you have? 64. That's a little bit, you know, extra. You don't really need those many, but that is why you have some redundancy. Multiple triplets code for the same amino acid. Okay. All right. Any ideas? Would you be able to write a computer program, very simple program, given a DNA sequence to find a gene, given the information of a gene that I've told you? Great. But is it enough to find a stop codon just later on somewhere? How would you need to stop, you're right, you have to find the stop codon, but you need to count in triplets, right? So the whole gene is always a multiple of 3. It's a 0 mod 3, going to the mod language, right? So it has to be that. So one of the things you could do is look for the first ATG, because this is DNA, right? And continue and in triplets, keep counting and look for the TG. This happens to be a gene, okay, that encodes a protein. Okay, and you should ensure that it's basically it is going to. That's one way of doing it. Okay, our DNA is really long, so you're going to have lots of false hits, right? By just doing this, you're going to see lots of ATGs, right? Just you can calculate the chance of that, right? Of finding an ATG arbitrarily. Right? And but this is one way, that is the first step, right? We're just looking at how to think about the problem. And this is the first step. Then you start adding things from biology and from maths that will help you figure out statistically robust ways to find genes and also to rank them, saying that this is something that I think is really a gene and so on, based on different criteria. So if you're interested in this, I can point you to many methods that try to do gene finding. Okay, all right. The other thing that can help a lot is the fact, again in biology, we are a product of evolution, right? And a lot of our genes are conserved, so across all the way to chicken, for example. Chicken, you know chicken, right? Yeah, hens. So lots of genes are conserved. They do similar things, the proteins, in different species. So one way to find out which region is a gene is to align different genome sequences with the human one. See which one looks like it's aligning properly. Right? So for example, this is going back to our genome browser. Right? Here's this region, human region, same region, aligned with a monkey. Okay, and you can see how similar things are. The ones that are not matching are just colored gray. Yeah? And you can do this with whatever genomes you have. And you can do this with mouse. Now you can see, right? We are further away from mouse than monkey, right? And that kind of makes sense. And you have a lot more. You see this? You have a gap, right, in the alignment. Okay, so what's happening here? There is either a deletion or an insertion, right, during evolution. Here you can see it's just changing, call it a substitution. Okay, so now if you're given these two huge chromosomes and you're asked to align them, how would you do it? You have a sense of what you're looking for, but you need to formulate it in math, using math to really say that this is my goal, right? So so first we need to define what is a good alignment. Is it better to introduce a gap here or to consider these guys as substitutions over there? You understand what I'm saying? When you're aligning two regions, this is done by a human or a machine, right? This is not the real genomic alignment. There's no genomic alignment, right? We are doing this. So now, should we introduce these 3 gaps to make sure that the rest of it aligns? Or should we just say that this G was over here matches with this G matches here, and forego the alignment later on? This is in our hands. We need to know what is a good alignment. Should we introduce a gap or not? Or should we introduce a substitution? Let me make this clear. So first, before you design any sort of alignment method, you should have some understanding of the biology. So you should understand what are mutations. Okay, suppose this is our original DNA. Remember, it's double-stranded. We always show one strand, but it's actually double-stranded. Now, during replication, or for some reason, there is a mutation. Okay, so this is a substitution. Okay, this G just got substituted with an A. Okay, and so the pairing got disrupted. Here is a deletion. So what happened here? Yeah, so this CG pair just disappeared. It was skipped while copying. Yeah? And here is an insertion, right? So maybe when the DNA polymerase that is making the copy in our cell read this twice and added something, right? These things happen routinely. Well, there is a rate for that happening, but these things happen, right? So all these things can happen. And when I said that these things happen routinely, we have a sense of which happens more often, right? So if we know beforehand that a substitution is a little more probable than a deletion or an insertion, we can have a scoring matrix that says that if my A aligns with an A, that's great. I'm going to give it a +1 score at that position. If I have to align an A with a C, so this is one genome, this is the other genome, okay? And this is just my matrix I'm going to use to score once I'm given an alignment. This is bad, right? But if I have to introduce a gap, that's really bad. So let's not introduce a gap as much as possible, but I have to look at the total score, right? And so you can develop methods to do this in good time, okay? And so a lot of computer scientists worked on this. They also worked on a slightly harder problem, which is to do a multiple alignment, not just pairs. Okay, so you see that now you're looking at a bigger problem where you have multiple genomes and you're trying to see overall what is conserved, what is not, right? And then you can just look at every column and sort of score that column, and you can come up with a graph. This is conserved, this is not conserved, and so on, right? Does that make sense? And this is hard to do by hand, by the way, okay? So you have to use a computer and you have to tell the computer exactly how to do it, okay? So that's why those gaps and all those things are important. And you have to use tricks to make sure the computer does it quickly. You don't want it to look at all possible combinations, okay? So if you like designing algorithms, I suggest you take some algorithms course, and there's some very neat tricks you can apply. Called dynamic programming that you can use for aligning these kind of for many such problems, okay? Which at the onset looks like it's really hard, but it's not, with some use of clever tricks. All right, so now let's see what's happening. So this is a real part of the real genome, human genome. You can see this is the gene. You can see how nicely conserved it is. You can zoom out, you can see that there are lots of patches. And if you didn't know anything about the genes, you could just look and say that this looks like there's something going on there. Maybe that is a gene. You could predict looking at the alignment and inferring. And over there, then you can see your ATG and go further in those things. This conservation score. So you're looking at the alignment and seeing how conserved every position is. And then it's just a height, right, of that, just a score to see how conserved it is. Here's a gene that's not conserved. Okay, so you can't just keep applying this blindly because we are different. So we're different for a reason. We're going to have some genes that are specific to us. So we can't just rely on everything that's conserved. So here's something that may have evolved from something, but it has changed. Yeah. So it's nothing fits, no answer fits everything. Okay, so you have to use different ways of getting to it. Okay, I can go on, right? Yeah. Okay. All right, so you have these different genomes, and you can now start figuring out where the genes are in each genome, right? And that can explain some of the differences that you see across different species, right? Because they're making different proteins. Right? And this can also explain differences within the same species. It might explain why some people become mathematicians, some become excellent sports persons, some become doctors, others start wars. right? So maybe there is some genetic basis for what we are, right? And that's good. So we're starting to get there. But what about just the same organism? So if you just look at all our cells, okay, my own cells, for example, okay, clearly my DNA is different from yours, right? Maybe at a few places, not like the monkey and human, right? It's going to be more similar than that, but there are going to be a few differences. But what about all the cells in me which have the same identical DNA, right? Or that's what they use, right, for all this forensics and all, because they know that the DNA is the same, right? So this is a nerve cell which looks really different than a muscle cell, than a bone cell, and so on, right? So when people didn't know, biologists didn't know much about genes and how regulation of genes happened, they thought that during differentiation, so during development, right, many of these things are sort of happen during development, right? Your bone becomes a bone, and etc., right? Your brain becomes a brain. They thought that the cells selectively, lost genes because they just didn't want them anymore. Because clearly the same genes are not active in all these cells, right? Our skin has melanin, right? Different amounts of melanin each person has. But now our eye lens, that is transparent. Now, if those cells decide, oh, I want to now start producing melanin, you're going to go blind, right? Because you need it to be transparent. So there's something that is making sure in our cells that although the recipe book is the same, that the paratha is not being made when the menu is Chinese, right? So something is doing that up there. Okay, although the book is the same, we have the same global book in our nucleus. Okay, so it turns out that, of course, there's a lot of variability of expression of genes. So if you just have these three genes, one gene might be really highly expressed, another gene must be low, and like this might be something that drives melanin is going to be really shut down. Okay? And another cell type, you might have a different combination of genes working. Okay, so it turns out that our cell is regulating all these steps. You remember the original picture I showed you with the cell nucleus and this thing going all the way down and creating the thing. All those steps are kind of being regulated by the cell, and the biggest regulation happens here at the transcriptional level, because if you do too much of transcription, then you're wasting energy, right? So if you don't want a particular protein, there's no real point in creating the RNA unless you think you're going to need it quickly later on. Okay, so there are reasons why you might want to keep it, but on the whole, if you don't see the need for that protein at all, the cell is just going to say, I never want it expressed. Yeah? Okay, so I told you that you have genes, right, in the DNA, and turns out that only 5% of our DNA is actually genes. Okay, so this gray part is actually 95% of the genome. Okay, this is different in bacteria. In bacteria, very little part of the genome is not coding for genes, hence the size difference. Okay, but still doesn't explain the onion part. I'll come to the onion part maybe later. Okay, so what are these other gray regions doing? Some of them are repeats. Okay, but what are they really doing? So it turns out that they are the ones that harbor signals that tell the gene to turn on or turn off. Okay, so it's like a software. Remember, our DNA is like a software, and it's the same when your operating system is doing an auto update. Right? There's something in the software itself that is telling you to do this now without you doing anything. So there's signals within the same operating system or same code that are driving this whole thing. So in coming back to biology, how is this happening? So I'm showing this pictorially as a straight line, but the DNA actually folds around a lot. Okay, and turns out that these regions, which I'm going to call regulatory regions, at some point that those words will come on the screen, hopefully. Yes? Regulatory regions. Okay, these are these regions. And the reason they're called regulatory regions and you see them being folded is because they are they happen to come together because of proteins, okay, which cause the RNA polymerase to come, which is like a signal to, okay, come here and start transcribing. And if all this works out, then your protein is going to be created or transcript is going to be created. Does that make sense? And this is also, by the way, a product of gene expression. It's like a chicken and egg. Who did this first? It all comes from when the organism was, created, was fertilized and so on. So from there, the journey continues. And you have these proteins, which are binding regions, and specifically, okay, so now here's where it is going to really recognize specific regions. You don't want random regions to be bound by the proteins because you don't want random proteins, random genes to be expressed. Okay, so you're going to have all this happening because of which the protein gets, the gene gets transcribed. That's called a binding site. Okay, what else do I have? Okay, so why are we interested in looking at the regulatory region? One thing is, of course, the example that I gave you about the melanin and things like that. Here's another reason, clear effect of a mutation within a regulatory region, not in a protein. Okay, so eye color is determined by a regulatory region, and that is because there is a gene that causes increase in melanin in the eye, not the lens part of it, the outside, right? And therefore you get this color in the eye, right? But there is a mutation that occurs in a regulatory region, not in the gene itself. So you can imagine that if the gene itself has a mutation, you've seen what a mutation is, like a substitution, insertion, whatever. Your subsequent protein that is created is going to get messed up because your code is all wrong, right? The genetic code is going to get messed up. You're going to put a different amino acid. So definitely, if there's something going on in your gene and the protein is going to be majorly affected because of that, it's obvious that you're going to have effects. But this is a perfectly good gene, but it is not being turned on when it should be because of a mutation in the binding site. Let me see if I have. So in this case, right, imagine that you have a mutation in this binding site here. That might cause this purple protein to not bind, which might cause this region to not come nearby, hypothetically, which might cause this RNA polymerase to not assemble, right? So these mutations also matter. So this is actually what we are interested in. This is what my lab does, to understand regulation of transcription. Okay, now I have to go through this set of animation. And so this can have an effect, what you really see. Now, I would like to say that this is just, of course, an effect that should not matter, but sadly, I think this color also may matter in real life as to your future and what happens to you. But you can imagine there are more serious effects, right? Like heart disease you might have because of a mutation in the regulatory region, which you don't know of. A gene mutation is very easy to catch because genes are easier to catch, right? We have all these methods to identify genes. So now after alignment, if you see there's a mutation in the gene, you can now figure out what might be going on. But the problem with these regulatory regions is that they're really, difficult to characterize. They can be far away. They can be, they're short. They don't have this nice structure of ATG, stop codon, etc. So there's no such thing, at least that we know of at this point. Okay, clearly the cell knows. The cell knows what signals are there and is working towards that. So it's really like searching for a needle in a haystack. Okay, all right. So I'll just quickly wrap up. So this is a small story from my postdoc. I'll just very quickly go through. So what do we have if we don't have any of this information, right? Or how do we get there? So what we did have is data. So we had some regions that biologists had painstakingly across years figured out that, okay, here's a regulatory region. I don't know what part of it is important, but here it is. If I remove this region, then I get, then I see a problem. right? We also have a set of regions which are not. So now we want to see, if you have this data, can I determine what it is within these two sets that make a regulatory region and the others not? Okay, so you have a positive set and a negative set. So if any of you have done any kind of machine learning, this is like that. Okay, this is something called supervised learning, where you know you have a label of two sets. If you've not done it in your coursework, I'm sure you have done it in real life. Okay, so although this is attributed to computer science, our brains are constantly doing this kind of a classification. Okay, so right from when we were babies, we can recognize the touch of somebody who is a caregiver and a stranger. Right, we are classifying based on features. We can't really see well. A baby cannot see very well, I'm told. Okay, all right. The other thing is that our brains are really good at this. So you have come across these kind of captchas, right? I'm sure you have. You're all social media people. You have to enter these things to get into any of these logins. And the reason they give us such problems is because computers can't solve them, supposedly, right? As well as us. Right? The reason for doing this is that they want whoever is running the server wants to make sure that whoever is entering is not a robot. It is not trying to just mess around with their server. It's actually a human. And so this is to detect that. Okay, I'm going to skip over this. So we are very good at those kind of images, but we're not good when we are given data like this, right? Because we've not seen such data from when we were kids. We know what ABCD looks like. We don't know what a regulatory region looks like. So what do we do then? If we have all these regions, positive and negative, we just do what we would now train a computer to do, to act like our brains. So the reason we can't do it is because we've not seen enough of them. The reason we can do such problems is because we've seen lots of mangoes and oranges, and we automatically detect features that are associated to each class. Yeah. So we want to make sure that we can build a computer algorithm that can figure out features for each class automatically without us telling them to, because we don't know what they are. Right? So what are the features we have? All the features we have are ACGT, right? That string. So we can just give it, for example, the number of A's, the number of AAs, and so on, or the frequency of this. And then you tell the computer, you figure out, you find the weights for every feature, and you come up with an algorithm that is going to decide what makes an enhancer and what makes a regulatory region a regulatory region. Okay, so we did that. And then once you have that, you have a classifier, okay, which is supervised learning. You just pass it over the genome and make new predictions, okay, and identify regulatory regions. So I will stop here. I think I'm just running out of time, but if you want to see biological experiments after this, which I did not conduct, collaborators conducted, I will show you later, but let's stop because we should not go over time. And if you have questions, you should ask. Good question. So it's often destroyed. So an RNA is very unstable unless it's going to be read again and again. So if it is a protein that needs, is like you saw the polymerase reading just going in the queue, right? Lots of polymerase molecules. You could have ribosomes making more copies if you need it a lot, but it doesn't hang around. It's very unstable. Unless the cell decides to and does special things. There's always unless, except. Yeah. Yes. So for this talk, it was continuous. But actually, for us, it's a lot of genes are broken up. Yes, yes. Do you know what these broken up genes are called? You're right. Yeah. So her point, what's your name? Anandi. Anandi is pointing out that our genes really contiguous the way I showed you. like starting from this word to another. Actually, in us, it's not. In higher organisms, it's not. So what happens is that the transcript is continuous, kind of, but parts of the gene or the transcript are just cut out and stitched. Okay, so it's a little. It gives us the ability to sort of do some kind of a combinatorial mix and match. Remove this part, don't remove that part. But then, so those regions, right, which are, it's called splicing. So you remove those regions, and then stitch back. That stitching is called splicing, right? So those regions have signals. So in your program, you can put something that is going to try to look for those signals. So even though you have those cuts in your gene, you do know a little bit about the signals that are associated, because this gene also needs, the cell also needs to know, no, that this is what should be cut out. So we are trying to find out what the cell is doing by looking for these signals and identifying. Yeah, good question.
If you remove that part from the gene itself, like, from the DNA, like,
Yeah.
will something happen?
Good question. So it would be specific to the gene. So sometimes they notice that some genes, always that region gets cut out. In other situations, that part may be retained. That is where this combinatorial thing happens. Like in one kind of a cell type, might say, I need that part to be coded for a protein. I need that little piece for the protein. The protein is like a complex structure, right? Maybe it has something jutting out, which it needs or doesn't need. And based on its need, it will decide whether to keep it or not. But a lot of genes, we don't see any evidence of it being retained, some of the introns, it's called, yeah. Okay, I'm here, yes.
Could you say something, and this is my complete ignorance about bio stuff, but could you say something about the COVID mRNA-based vaccine? And is that related to how it actually goes in and sort of interacts with the proteins, or is that something